Let's check this out. All right, before we get started with chapter 6, which is going to start us in with genetics, and it's a pretty heavy chapter. This and chapter 10 are probably the heaviest in this unit, so keep that in mind. But before we get started with this, anybody have any questions about anything, about questions that got wrong, or I don't know, whatever else? Okay, fair enough. What else do I want to say? I feel like I already went over everything with you guys after the test, so we really need to go anything. So we can just jump into this, I guess. We don't have a Kahoot this morning, of course, because we're starting a new unit. So this will be a day where we get that extra time. I'm not sure that we wanted to spend that extra time doing lecture, but that's what we're doing. So we'll need it for this chapter, unfortunately. So when we get into genetics, we often hear this term about, an organism's genome and chromosomes and everything like that. So the genome of an organism, technically it refers to everything genetic in the cell. So all of the genes, whether it's part of the chromosome or not. So consider this. We talked about mitochondria and how they have their own DNA. Their genes are not encoded by your DNA, right? They're only in the mitochondria. So if we consider our chromosomes and the genes of the mitochondria in our cells, that's going to be the genome for those cells. That's just an example, right? Anytime we have any of these bacteria that, they may have incorporated viruses into their genes, into their little circular chromosome, that's easy enough to include that in a genome. But if they have plasmids, those little round extra pieces that can have like antibiotic resistance and they can transmit it between one another, that's also going to be considered part of the genome because it's going to be anything genetic in the entire cell. So remember that cells will have DNA always for their genome. Viruses can have either one. Now, do we have RNA in our cells in general? Yes, we do, but they're not going to be our genome, right? They're going to be making copies of the genes that are DNA. So that's not the case with viruses. Viruses, they have it as like their main copy. All right, so a chromosome is a distinct cellular structure composed of neatly packaged DNA. We kind of know this, we're used to seeing this. As we had described before, in the terms of mitosis, right? So when we think of mitosis, we think of everything being in those X's, right? And it's actually two chromosomes connected to each other at the centromere. That's what that's called. You don't need to know it, but there it is. There's a term for it. Anyways, and you'll have basically, like if this was your set number 1, chromosome 1, you'll have 2 of them, right? You'll have one from the mother and one from the father, genetically speaking, okay? And we have copies made of them so that whenever we go through mitosis, we can separate like this guy into one cell and that guy into another cell, and the same thing with this. So that those each go into the daughter cell. So the daughter cell will end up having one of the mother, one of the father, one of the mother, one of the father. Right, so that's mitosis. So that way we can keep the same number of chromosomes whenever we divide, because our cells do not exist like this every time. Like that's not what they normally do. It's only when they are replicating and dividing. We're going to learn about that replication today. And I want to keep in mind that replication is something completely different than transcription and translation. A lot of people get confused, so I just want to head that off really quickly. And to set this up so that hopefully we can keep everything in our minds in the order that we're about to learn it, we will have DNA making a copy of DNA, right? And that would be what we had to do to get these, yeah? So in order to get those singles, we're making this into a new copy of a cell, then this would have to double up the M and double up the F in order to separate them so we keep the same number, right? So in order to get that though, you have to duplicate or replicate your DNA that you have, all of it, and all the chromosomes. So all of our 23 pairs, right, so 46. But anyways, so this process of making the copy of the DNA This process is called replication. Makes sense because we're replicating the DNA. But all of these are like Asians and stuff, so I don't want you to get lost in the terms, okay? So that's replication, easy enough. If you wanted to go from DNA to, you guys know this process now, we mentioned it briefly, to RNA into protein, which is the whole point of your genes. If you're wanting to go through these processes, going from DNA to RNA, this process is called transcription. And this, usually if we're making the messenger RNA our working copy, I'll put that here, mRNA. This is our working copy of the gene. You see it, well, it's up to we learn all this stuff. This is also known as the transcript. Right, I didn't mess that up at all. Okay, sure, that's very good. And then if we're going from RNA to protein, right? So that process is something else. So I'm going to circle these so we can see these terms because these are like our biggies for this chapter. So you can figure out where we are in the process. Okay, this one doesn't need to be circled. You'll know that this is part of that. This is called translation. Did it again. Then I want to point out to you guys, so that's how we get our protein. You know, proteins, they're made up of amino acids, right? Let's put that here just to remind everybody. Okay. So if we were to look at this, DNA to RNA to protein, this part of it, we were to look at what DNA is and what RNA is. These guys are both called nucleic acids, right? Easy enough. That's not news. Shouldn't be news. Learn this in our macromolecules, right? So those are nucleic acids. It's in the Na. That's literally what we're going to be talking about. So deoxyribonucleic acid, ribonucleic acid. So it's there in the actual abbreviation, so you know what it is. Obviously, protein is something else entirely, right? We're kind of talking about something written in the form of amino acids. Right? So these are technically in the nucleic acid language. I would see it that way. Whereas the protein language, so nucleic acids for these guys in the name, fine. Proteins, we'll just put amino acids over here. No. Okay. So whenever you are transcribing, think of it this way. If you're a scribe, literally like back in the olden days, like in the Renaissance or whatever, and you have this, one of these books that was like the gilded gold books that they have, right? And they did all of that by hand. And if you're looking at that, you just have to make a copy of something from that in order to bring it like to your, church that you're starting up and forming, on your land or whatever it is. You would be a scribe that would be transcribing that. It's the same language. Scribes are writing, right? We can agree on this. So scribes are going to be writing things. So just think of it as that's the same language. Because I think we can also agree that translating something requires going into a new, different language. So this is clearly a different language. So if anything, hopefully that thinking can help you guys keep those straight. transcription versus translation. They both start with T, so you know they're going to be involved in that whole process of creating a protein. The first one's going to be transcription, the next one's going to be translation. You want to copy down the gene, the part of the book that you're interested in, and then you're going to translate it into a different language after that, right? So that's what we're doing. All right, so that's the concept of what we're about to get into with a lot of this stuff, and I wanted to put it up there. I know that doesn't seem basic, but it will when we get into this, and I just didn't want you guys to get lost in the terminology. So we did this, yes. Okay, so bacterial chromosomes, they're typically going to be like one circle. They have one circle. They are still double-stranded DNA. It's just in a big old circle. And that's going to be it. Ours, we have obviously multiple chromosomes. We have one from the mother and one from the father in our cells. Not all animals work that way. They can have whatever combination that they feel like in their, I don't care kind of stuff. But for us, and for most eukaryotes, you're going to have one from the mother and one from the father. So, and of course, these are just euphemisms for who's donating the genetic information. So we are considered as humans to be diploid because we have 2 of every chromosome set. So this, when I'm talking about this up here, when we get into genetics, not just how this stuff is made, the actual genetics. This stuff here is what's going to be determining basically what's encoded in your cells, right? But all the cells on the mother one and all the genes on the mother one and all the genes on the father one, they're all the same genes, but they might be different versions of that gene. So one of the examples I'm going to give here in a moment, think of it as the brown hair versus the blonde hair gene, things like that. Or curly hair versus straight hair, or green eyes versus blue eyes. These are all easy ones that we can see on people, whether we're talking about their height. There's so many genes involved in so many of these things. It's not always that simple, but you can understand that that one chromosome is going to have the same gene. So this will have eyes, hair, height, whatever is on there, right? So they all have the same genes. Maybe you got from your father blonde hair and your mother brown hair. I don't know. And then based on whether those genes are, and I'm sure you've heard these terms, dominant or recessive, determines which one's going to be expressed. And it has everything to do with how your body's going to be regulating the genes to determine which one's going to be going through this process here. It's way more complicated than you'll have to learn. But that's how this is, that's the importance of all of this. So we have 3 things that your DNA encodes. Literally, these are it, okay? There are structural genes that code for actual proteins, the genes that code for the RNA machinery that's going to be helping with this. Here we're talking about things like ribosomes, which are made of RNA, ribosomal RNA, and transfer RNA. We'll get into what they do in a moment. And then we have regulatory genes that actually control the gene expression, ways that you can turn the genes on and off, or how much you need made and stuff like that, okay? But in case you haven't noticed, the real output of your genes is going to be proteins, very largely so. That's all your genes code for. You don't have anything else they're coding for. All right. So we talked about the genotype. Well, we talked about the genes that you could get from your mom and your dad, right? And let's say from your mother, you had gotten a chromosome that was for brown hair, and then the father was for blonde hair. I'm going to write them like this because they're both the same gene. They're just different versions of that gene. So we're using B to demonstrate that gene, hair color, okay? We would say big B because it's dominant, and little b because it's recessive. That's typically how we're going to write it. So this will be just to introduce the concept, okay? So dominant, and this is recessive. What dominant means is that it will be favored in going through that DNA RNA protein process. So typically that's the gene that's going to be turned on. You can have genes that can be turned on in some tissues, and then in other tissues they'll be turned off. Obviously, if we had all of the genes turned on in all of the tissues, we wouldn't have tissues, would we? So we have to regulate based on that. But you can even get even more complicated. If you look at somebody who has heterochromia, we're talking about expression of different genes in their eyes. If we're talking about a calico cat has their like fur color coded well, I will say this, the control of their fur color coded in their X chromosomes. It's a recessive trait. So you have to have two of it in order for it to really make a difference. And so that's why we're going to be only seeing that really in female cats. In case you didn't know that, there are no male calico cats. So that's interesting how stuff like that'll work, right? We also have diseases like SCID, which if you don't know what that is, that's severe combined immunodeficiency. So it's boy in the bubble disease. So these are individuals who have, and this reason it's called boy in the bubble, not person in the bubble, okay? These are genetic defects that make it so that this individual doesn't have an immune system. I mean, effectively none, no immune system, not even the basic immune system. So they can't be exposed to the outside world because they have absolutely no way to defend against it with their own body. And that's why they tend to die young, because even in a bubble, you can only control it so much, right? So having said that, that is a trait that is carried on the X chromosome. And now that is on the X chromosome, that is recessive. So that has you thinking like, well, that's a recessive trait. Actually, let me draw this on the thing so you can see up here. Okay, that's a recessive trait. So like, let's say little b, let's just use that same thing, right? And it was on your X chromosome. So here's your X chromosome. And if you're female, you're XX. And if you're a male, you're XY. So if you're a female, you have the opportunity to have two versions of B, let's say. So you would have like B and little b, for example, but you'd be only seeing the expression of the big one because it's dominant. However, if you have this, you're not carrying B's on there. Y's, in case you don't know, carry completely different genes from X's, not the same. So males depend entirely on the X chromosome, the only other chromosome it has in that last pair, for the genes on the X, since they're not going to have a duplicate of it in the Y. They got the Y from the dad and the X from the mom. And that's what they got. That's the only X they have. So if you have a gene that you happened to get from your parent that is little b on this, you don't have a dominant. That's what you'll express no matter what. And if you look at it this way, if you are a male and if you're carrying that little b with you and you have to express it, then you will always have SCID. And how many times do you think that's going to get passed on to offspring? Like 0. Yeah. I mean, that person doesn't even get a chance to have a child, probably. They're kept in a bubble or they died. So that's the reality of the situation. So probably not passing on that gene. So what is the issue here? Like, why would we even be worried about it if that is the case? Well, if somebody comes along, with some sort of mutation that can lead to that, that could be an issue. And there's a bunch of factors that can lead to it eventually, but that's why it's rare, right? We don't see this a lot. It's super rare. And there's a reason for that, because it's all linked to this, and because the males that have it are going to die off and not be spreading that gene. And it tends to be mutation related. So, okay, so that's the big brains stuff. Let's go back to the brown hair and blonde hair thing, okay? So let's look at this. We'll get to genotype and phenotype with this. Let's say your mom carries 2 genes for brown hair, all right? Her mom, grandma had brown hair gene, and grandpa donated brown hair gene to mama, okay? So this is your mama's genetic makeup, and whoever you were getting father, whatever genetics from, goes over here. And let's see that that person, the father, I didn't do myself enough space, will have a big B. So they have brown hair, but in their family, maybe in his mother's side of the family, they carry blonde hair gene. Now they both look like they have big B and little b, okay? So that's literally B looks like this. I know it's over the letters, so I'm trying to write it so you can see it. So this is something we call a Punnett square. You don't have to do these, but I feel like it demonstrates pretty clearly the different possibilities of offspring you could have if you were looking at this. So even though both parents look like they have brown hair, they can have the following offspring. You would have big B, so we're just bringing down or across these guys, okay? You could have, so big B across, little b down. You go ahead and bring that little b down here, and then big B down to this big B. Yes. Okay. Yeah, these are pretty common ones that we can get in biology. We go through these are pretty simple ones, right? So if we look here, all of them have a big B in front of them, right? So if we look at this particular genetic situation, we'll only have children with brown hair, only. No opportunity for expressing that blonde gene. However, 50% of your children will carry the blonde gene, right? So what happens if we were to have a mix up here? Let's say both of them were big B, little b. This is the typical setup that we would see to give some variation. So here we have big B, big B, big B, little b. Like I said, I'm not going to make you guys do these, but I feel like this helps demonstrate like why things are rare or uncommon or whatever. So only 1 in 4 chance every time you conceive, not in all of your children. It's not like if you knock out a kid, you pop out one kid and that kid had, you know, brown hair, and now you have 3 chances left and one of them's going to be, that's not how it works. Every single time that you conceive, there is a 1 in 4 chance that they will carry the blonde, express blonde, the recessive blonde. But you have to have little b to get that. So 75% chance every time that you conceive that kid would have brown hair, for example. Now, this is one gene we're looking at, right? Imagine how complicated it gets. And that's why where we get all our variation from person to person. Not to mention, there's some genetic switching up as well. So anyways, that's the basics of genetics and why it works the way that it does. So your genotype would be like big B, little b, right? That's your genotype. You have one from your mom is the big B, one from the dad is the little b. Your phenotype is what's expressed. So this would be brown. Or if you were little b, blonde. Make sense? So phenotype is what's expressed. Your genotype is what your exact genes are going to be, mom and dad wise. Okay? Whether you're seeing them or not. You'll be carrying genes sometimes that aren't expressed. All of us are actually right now, somewhere in there. So anyways, there you go. That's the idea of those differences. We know what DNA is. We know that it's going to be DNA and RNA are both made of nucleotides that are going to, consist of some sort of pentose sugar with a phosphate and a nitrogenous base. In the case of DNA, we're talking about deoxyribose sugar. And then we have our nitrogenous bases, adenine, thymine, guanine, and cytosine. Like I've been saying to you guys, I'm not going to make you pick on the exam, maybe on quizzes to get you guys looking because y'all can use open books on quizzes. But on the exam, I won't have you picking between like adenine and like alanine. Those are no, that's ridiculous. So I won't make you know that. That's not what your job is. So, but A and T always bind together, and G and C always bind. It's never anything but that. So G and C, I remember that G and C are both like shaped the same. They're both round. And so those are the guys that would be binding together versus A and T. They have the crossbar effects going on. That's how I remember it. So whatever works for you, but A, T, and CG. Always going to be what binds. All right, so when we look at DNA, we're looking at an anti-parallel arrangement. This is very hard for me to describe. So there's a picture on the next page that I'm going to show you guys of what I mean by this. But the fact that each side of the helix, each side of the ladder, we're talking about a DNA ladder like this. You've got the rungs, and of course it's going to be G to what C and whatever all the way down in the middle. But each side of this ladder runs in a different direction. So this one would run this way and that one would run that way. So that's anti-parallel. Now let me show you what that means chemically, because if I try to draw it, it's just going to get confusing. You see what I mean? So, okay. You guys remember in chemistry, please tell me you at least vaguely recall this, that you will number the carbons in a sugar based on like their position around the ring and whether they have something coming off of them. Please tell me that you understand that, because if you don't, this is going to make absolutely no sense to you. So you're going to number your carbons around whatever your sugar is based on the position of that carbon, whether there's oxygens there first or whatever. And I'm not going to make you number them or anything. I'm not going to make you call out which one is numbered or anything, but this all depends on that. So everything is going to be oriented. I want you to always think of everything as 5' to 3'. And what that means is that that number 5 carbon out here versus the number 3 carbon out here. How they were numbered structurally. And you don't have to draw them or know where those are. But so that you know what it means, it's referring to the sugars. So you'll see on the other side, it is literally flipped the other way. Here's your 5 carbon, and we're ending in our 3. So it's whatever is at that end, basically. Is the 5 carbon there, or is the 3 carbon there at the end? So those are the directions. You can see now that these are, in fact, inverted from one another. Now, that's just the backbones, okay? The backbones are inverted. So in all of our backbones in our DNA, and you can see it here, so I'm not going to draw on top of it because it's nice and clear there, but you will have phosphate bound to sugar, and then phosphate bound to sugar. And of course, it does matter where the carbons are in this, and that's where we get the 5' and 3' from. But both of them are going to be alternating phosphate, sugar, and that's where we get a nucleotide from, which because phosphate to sugar, always off of the sugar, you can see that in this diagram. It's not coming off of the phosphate, it's coming off of the sugar, the adenine or the thymine or the guanine or the cytosine. And they look ridiculous. They look like these things that I'm also not going to have you draw or recognize. Okay, so that's what's going on. And then they will create hydrogen bond attractions to the guy across. So one of them has nice, happy, positive hydrogens, and the other one has nice, angry, negative oxygens. So the hydrogens and the oxygens getting attracted to each other across the way, creating hydrogen bonds. That's exactly what we were talking about. Now we're getting down to it at the chemical level and not just saying they're held together by hydrogen bonds, but now we're seeing why. Okay, so that's why we call it anti-parallel. They're running alongside one another, but they're running in different directions, the backbone of it. Now you can see here, if we were to write out a genetic sequence of DNA, if I were to have a sequence that read something like A, C, G, or whatever, and I know this would be definitely your 5' and your 3', and then your other side would have to be written like this. And how would we write that? We'd write it T, T, G, C, because A always binds with T, and G always binds with C. And that's where, if I simplify it down, you can see a little bit better that how that hydrogen bond situation is going to work, and then in respect to that anti-parallel. Okay, but always remember that the whole nucleotide is there. The phosphate, the sugar, and the nitrogenous base. And they are what's repeating over and over again. It's just in what orientation. Yes, I know. It's really fun, isn't it? So, so when we make a copy of our DNA, we make it semi-conservatively. So what semi-conservative means is that we will take, this is actually better if I do write this with a marker, because I think I can show different colors with it. What's a proper different color? We have what, black over here, so this purple, maybe this will show me this one. Oh, brown, it's brown. Black, let's do it over here. Sorry, I apologize for those of you who can't really entirely see it, but if this was, I'll try to draw it up here high so we can all see. Your original DNA strand, right? And we have this is where your A to T and G to C is happening, right? And we have anti-parallel. This one's 5' to 3', this one's 5' to 3'. Force. Okay, that's what's set up as. What happens is you're making a copy of this. You'll split this guy apart. We've kind of already talked about this, the concept of this, but this is what would happen. You'll split these apart to make a copy. This is still in the same orientations that they were in. And then we're going to add nucleotides to each of those, using each of them as a template, okay? So think of red as old. Here, it's original, right? Original. So we would add, I don't know, A to T and G to C all the way across here, and then have that be our next new strand. And guess what? Of course, that's going to be in the correct orientation. Same thing with this one. Adding our nucleotides. Okay. So do you see how the new strands, we'll put a plus here, so you know that there are 2 new strands from the one, consist of old and new strand, right? So the new ones We have an old strand that was a template to build the new strand off of it. So that old plus new, and that being the same every time if you split this apart, then they'd both be old, right? And so on and so forth. But old, and then always half of it be old and half of it be new. This is semi-conservative. We've conserved one side, but not both sides. It's not, we didn't conserve all of it. Only one strand. Every time. One strand's the old strand, one strand's the new strand. This, of course, is not at all probably showing you anything helpful in that regard, but it is going to be helpful for what we're going to look at soon. Okay, so that each one is, neither one is completely new, neither one is completely old, right? Half and half. All right, DNA polymerase. I'm not going to have you remember that it has to be DNA polymerase 3 or anything. DNA polymerase, we talk about a polymerase. This is an enzyme, ACE, right? That is going to polymerize or make multiple things come together, DNA. So literally taking DNA nucleic acids and linking them together to make DNA. So that's what that enzyme is going to do for us to create our new daughter strand. It's the one that's going to be adding this on to make sure it matches up. Making sure that A binds with T and T with A and whatever, you get it, right? So it's going to be adding those on appropriately, but it can only add on to an existing chain. If this were the actual beginning of the chain here, that couldn't add on from here. Why? Because it's not anything on this end. So that comes into play as well. So we tend to use a primer when we're doing this. If you're starting from scratch, then you'll need a primer first, and that's usually an RNA primer. And we can trash it later and replace it with DNA. Because we'll already have DNA there, we can add it on from there. Anyways, and it only, and this is why we tell you about 5' to 3' and why you need to know it. It only adds nucleotides in the 5' to 3' direction. Okay, so that means if we had looked at the first strand up here, not even looking at the bottom one, okay? If you were adding on nucleotides in the 5' to 3' direction, we're talking about the nucleotides moving that direction. So you'd have to start from this end and move that way. For this other one on the bottom, this end and move that way. It can only move 5' to 3'. That's why I always say it, 5' to 3', so hopefully that will stick in your lines when you go to the test. And you're like, okay, it's always 5' to 3'. She never even said the other one. So what we do is we have a lot of DNA typically going on, whether we're a bacteria or a eukaryote, it doesn't matter. We have a lot to deal with when you're having to replicate it, right? So we start usually in a bubble area, and we'll call that the replication fork. And it's where we're going to separate the DNA, because we're not going to separate all of it all at once, because that would be nuts. That would be a lot of DNA just floating separated out there. It could be dangerous for trying to keep everything together. So we separate it gradually, and we open it gradually, and we close it back behind us as we're going. So this is the replication fork, where everything's going to be going down. And eventually we'll have the whole chromosome done from there. And we're going around. Again, we need primers, RNA primers to start. Okay, so leading strands and lagging strands. This one is almost impossible for me to describe. So I don't think there's a good picture of it. No, I'll try. Okay, if you have to add your nucleotides in the 5' to 3' direction, and you're working at a replication fork, and that's what this was. The picture over here, that's a replication fork, okay? So remember that we'll be pushing in both directions. We'll be pushing that way and that way to eventually make copies of the whole thing. So at one end of that replication fork, you are adding nucleotides 5' to 3'. And you're slowly opening up more and more and more and more as you push in, unwinding that DNA and pushing in with that polymerase and adding on the nucleotides. Seems fair, right? 5' to 3'. The problem is that as you're opening up more of this empty side over here. And that can't make copies this way like the other strand is. It can't just keep going. It can only add in 5' to 3'. It still has to follow the chemistry rules. So what we do is we make it in parts. We'll open it up a little way, and then we'll add a little bit on there. We'll open it up a little way, and then in this area, we'll be adding a little bit on there. So we'll make this in fragments. So the strand that can make it all at once, this is called the leading strand. Makes sense because it's the one going straight on through. It's basically leading the way. This strand over here, it's made in parts, in fragments. This is called the lagging strand because it has to kind of catch up a little bit. We open up a little ways before we can start on that side. This is the fallacy or the problem that we might see with having to go 5' to 3', but we found a way around it. Our cells did anyways, okay? So we'll make it in fragments. These fragments that we're making, these little pieces, we'll eventually join them together with an enzyme called DNA ligase. If you don't know, this is referring to ligation. To ligate something is to bring it together. So this is, I want to say, it kind of seals it up. All right, and then these fragments that we need to seal back together and that we have to make in fragments because isn't it fun on our lagging strand? These are called Okazaki fragments, obviously named after a guy who discovered about it. Okay, so that's leading strand, lagging strand, and Okazaki fragments and what we need to know about them, okay? All right, so now that we definitely understand DNA replication, which is what that was, making our copies of our DNA, we've got the leading strand pushing in and the lagging strand getting gradually opened up and making it in fragments as it's opened up, okay? And then sealing it together with DNA ligase. And replacing any primers with DNA. Okay, so now we can start thinking about how transcription and translation are going to be dealing with our DNA. So we consider that to be our central dogma, DNA to RNA to protein. We kind of talk about that. I'm not too concerned about what it's going to talk about, the problems with it. And there's different kinds of RNA that we can get out of here. So our main one that we always like to think of is mRNA. This is our working copy of a gene. The one we're going to make the protein from. We're going to translate that message, okay? This tRNA, I definitely wrote over it so you couldn't see it. tRNA, this guy is going to carry over the correct amino acids. And then this nonsense, see that shape of that thing? This nonsense is ribosomal RNA. Ribosomal RNA, like it says in the name, that's going to be dealing with, the structure of the ribosome. It's the main portion of it. Okay, so for everything that we're worried about for this, ribosomes are made up of ribosomal RNA, and don't worry about anything else, okay? Because they're largely that, and I feel like it's important to understand that RNA has more functions than just messenger. We can also make little copies of, like, let's say, part of a gene that just matches that gene's sequence, and take that to help silence genes. And that's what microRNAs and things like that do. They just like help keep it from making transcripts and stuff like that. They just bind there and block it and stuff. We're not going to get too far into that, but it's kind of interesting that they exist, I guess. All right, if you were trying to look at all the proteins that are made, because remember I said some of your genes will be expressed in some tissues and some in other tissues, and some of them are dominant, some of them are recessive, and some people or organisms have that expressed differently depending on their situations, right? Proteomics is the study of the proteins made in an organism. Just that's what that is. So, you learned it. Okay, so this is how the DNA to protein situation is going to go. In DNA, this is our double strand, right? If we were to look at this messenger RNA, this is the messenger RNA. So this is double-stranded DNA. And you would look at the bottom strand up here, this one here, of the DNA, and then compare it to what you see in the shapes below it in the messenger RNA. It's the same exact thing, right? It's the same. So we would call this the coding strand of DNA because this is what is carrying the same code as what's going to be on the messenger RNA. However, we didn't use that as our template, obviously, because if we were making a template and making a copy, you don't follow the A, T, T, G to G, C to C. No, we have to go opposites. So that's why this top strand is the template. That's what we use to make the messenger RNA. So it's the template. The coding strand is like what would carry the message essentially if you were to just read it. Okay, so we've got that. So we've got our messenger RNA. Again, this is our working copy of a gene. And remember, your chromosomes are like massive and you have multiple of them. And so you're just trying to go in and get info for one gene. So we're not needing to go through the whole of every DNA that you have, okay? Anyways, while we're going from that working copy of the messenger RNA, we're then going to be making proteins, which are essentially amino acids linked together, right? So this 3 nucleotide code here is called a triplet on the DNA. Triplet, okay? I'm going to erase this because I wrote over the other one. So this is the triplets. Come on, dude. All right, on the mRNA, this 3 nucleotide sequence is called a codon. One of the most important terms you're going to learn in this chapter. Because a lot of the questions are going to ask about blah, blah, blah codon. And if you don't know what that's talking about, you're going to feel pretty lost in a lot of it, okay? So it's our codon. It's a 3, always 3, always 3, always 3. Never any other number, okay? Always 3. It's 3 nucleotides, 3, 3, 3, 3, 3, 3, 3, all the way down. And you could say that about the whole DNA, but we're interested in the mRNA because that's what's giving us our actual protein. So our 3 nucleotide codon sequence codes for an amino acid. The next 3 codes for a different amino acid. The next 3 codes for a different amino acid, and so on and so forth. And that's where we're going to get our amino acids from to build our protein. We'll walk through how that works, of course, in more detail as we go through it. So in our transcription and translation, we've already introduced the concept of the messenger RNA being a working copy. Transfer RNA is going to carry our amino acid, and that's what we're looking at here, over here in the picture. The amino acid will attach up here. We'll have the regulatory RNA as well. We're not really going to get into that. The ribosomes that are made up of ribosomal RNA, which is also our RNA. So I wish that this hadn't been here, but I didn't make that slide, so I forgot to take it out. So this ribosomal RNA, only our RNA you have to worry about is ribosomal. And then different kinds of enzymes that are involved and the raw materials that are going to be going into this and blah, blah, blah. So there's a lot going on. All right, so in RNA structure, typically we're going to see RNA as single-stranded. Most of the time, messenger RNA is single-stranded. It's just one strand carrying that message. It will also exist in a helical form. It's just the nature of how that chemistry of it is. And then it will also have uracil instead of thymine. So we have U instead of T. But they're the same coding effect, okay? It's just we're not using T in RNA, we're using U. Again, that has to do with the chemistry of how it's going to work later. And we're going to use ribose as our sugar rather than deoxyribose. That's kind of self-explanatory in their names. So those are our differences. We already kind of talked about these guys. We don't need to talk about that again. So on the transfer RNA, the way that that transfer RNA, that upside down T looking guy, is going to identify which nucleic or which amino acid to add based on the codons of the messenger RNA. So if you have, you know, nucleotide, 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 3, 3, 3, this transfer RNA has another that matches up to it. So if your sequence was, let's say, G, G, C on your messenger RNA, the transfer RNA will come up with its little piece that looks like a little butt at the bottom of it, and that would match up C, C, G. And this is how they recognize one another and know that we're getting the right amino acid adding on. Okay, so it's literally based on the genetic code. So these 3 nucleotides will code for a specific amino acid. This is called the anticodon on the tRNA. And this is called the codon on the mRNA. All right. We'll get pictures, no worries. So here's the ribosome. Again, for our worries, it's basically 2 big units that are going to clamp down on our messenger RNA. And it's made up of ribosomal RNA. Okay. And it has these little pockets you can see where we can have transfer RNAs come in, this little tRNA upside down T guys carrying our amino acids, coming in and associating with the messenger RNA in that pocket. And I'll walk you through how that's going to actually make the whole thing work as we go through that. But we have to still learn about transcription. That's the whole point of what we're trying to get to is this protein. So transcription, remember, DNA to RNA. To make that messenger RNA first, we have to go through transcription, right? Making our copy of the gene as a scribe. And remember, you can't take the DNA out, but you're going to be doing everything else with it in the cytoplasm. So you need something to take with you. So this messenger RNA, we're going to make it in 3 steps, and it's easy. The first step is initiation. All that means is that we have, go figure, this is RNA polymerase. Should give it away what it does. RNA polymerase is going to recognize a sequence on where your gene is called the promoter. It's always going to be a promoter there. And it'll bind on that promoter when the cell allows it to make that messenger RNA you're going to make. And then that's initiation, basically. Binding of your RNA polymerase at the promoter, and then now starting to separate the strands to get ready for building. Next step, again, there's only 3. Next step is elongation. We're just going to be adding our nucleotides, 5' to 3' direction, to start with that 5' on our new end, going all the way down. Okay, so 5' to 3', adding of nucleotides. You only need a single strand because we're an mRNA, so we'll just be making that copy from our template strand. Cranking along, adding nucleotides, however they match up. Remember, RNA is going to have U, so it'll be, if it said A on the DNA, it will be a U on the RNA, okay, not T. But anyways, once you get through the whole thing, there's a signal for the RNA polymerase to dissociate. And that would be our termination signal. And then that will kick off that RNA polymerase and then release the messenger RNA. And then we can take that out into the cytoplasm, to the rough ER, for example. Okay. 3 steps: initiation, elongation, termination. Turns out we have the same steps for the other one. Where are we time-wise? Trying to decide if I really want to do the questions right now or not. If I want to go through, because it's a lot of info, but do you guys want to do questions or no questions? Not questions, I get it. Okay. We'll walk through it. So what's the 3 parts of a DNA nucleotide? Phosphate, sugar, nitrogenous base. So in this case, deoxyribose, right? What makes DNA replication semi-conservative? We're going to be separating strands, and the daughter strands won't be all new. They won't be, all old. Each one will be a mix of both. All right, during DNA replication, which strand makes Okazaki fragments? The term for that strand is the lagging strand. Generally speaking, what is the overall purpose of transcription? We're trying to make a working copy of a gene to take it out into the cytoplasm to make protein. It's our template for that protein. And what is the function of messenger RNA? Again, also, it's the template of the protein. So it'll have the codons that code for specific amino acids. And that's what's going to build our proteins. Okay, so start with all this. I don't know what else to say other than the codons. We've gone over it enough, I feel like, at this point. But 3 nucleotides. Oh yeah, this is better. Okay, good. I didn't know this was coming up. All right, so let's say, I keep telling you guys it's the 3 nucleotides that make the difference, and it's always 3, and that's what's important, right? If your mRNA says U, then that codes for phenylalanine, for example. But if you look real quick, UUC also codes for phenylalanine. So there's some overlap in these guys, generally speaking, right? You look through, we have 20 amino acids here that are typically used by most living organisms. And then we have some of them, UAA, UAG, and UGA, that say stop. They're stopped because they don't code for an amino acid. They're not going to call for a tRNA. And when that happens in the ribosome disassociates. That's what I want to say, but that sounds like I'm talking about psychology or something. But it disassembles, let's say. It comes apart when it sees that. And yeah. So I feel like the only one I'm seeing on here for our actual amino acids, there's 2 of them, that are coded by only one, and that's going to be tryptophan, which is our UGG, and our AUG is methionine. And that's the only ones. Everybody else has at least two. But anyways, there's a reason for that. And you'll notice that it tends to be the last position where we'll have that variability. So UUU to UUC. That's because of something we call the wobble effect. So that if your genes coded UUU and you had a mutation and it became UUC, you're still coding for phenylalanine and it protects your protein structure. And that's kind of what that's for. Yeah. So obviously some of these, it tries to protect more than others, but you get it. So here's a nice little overall picture of all the stuff we're learning about. Our DNA triplets up here at the top, we have the coding strand. That's going to have the same as the mRNA, essentially. Just replace that T with a U. And this is the template that we are using. That we will use complementary RNA nucleotides to build. So here's our, remember these are called triplets. These are called codons. And these are called anticodons. They are going to be up here at this end. You see how those will match up? A to U, U to A, G to C, and so on and so forth, right? And that's how they're going to recognize one another. And then these amino acids that are attached to their respective thing that's matching up their anticodon to the codons that was coded for in the triplet. This is the amino acid that's going to go in that protein. They always start with AUG, and they always start with, the protein will always start with methionine because of that. So F-methionine, I believe, is what we use in bacteria. And I think humans use just methionine or some other version of it, but it's always the start. And sometimes they'll clip it off when we're done, but it always has to start with that. All the elements for synthesizing protein. So yes, we'll bring all of these together. Basically, the messenger RNA will get clamped down on by the ribosome parts. There's two parts, right? And then that will start the initiation procedure. Our start, like I said, start is always AUG. Stop just does not code for an amino acid. So that's why we just separate from there. The translocation is what we're going to do while we shift down. Now, there are videos of a lot of these processes that I'm trying to describe to you with drawings. The videos, I can't act out thing, right? So I do the best I can, but I can't do that well of a job as the videos. I highly encourage you guys to watch the videos at some point while you're studying or something. They were what came in that announcement. So let's just get a chance, especially for translation, because I think it's one of the coolest processes that your cell can do. So our ribosome is going to clamp on down onto the messenger RNA, and we're going to start. We're going to bring our AUGs are going to be our codon, and the anticodon, the UAC on that tRNA is going to bring in that methionine. Great. The next little slot next to it reads the next one. So this one, in case, this is going to be CUG matching up with GAC on the anticodon, and that's going to be carrying leucine. All right, so cool enough, right? We're going to create, like it says down here, a peptide bond between the first and the second amino acid. Once that happens, we can release the first empty tRNA. It's not attached. It doesn't need to be attached anymore. So we'll release that guy. Okay. Then once that guy is released, we can translocate. This is our first translocation. Now our 2 guy is in the 1 spot, and the next guy can come in, and we'll bond him, and then etc. and so on and so forth. We'll keep moving on down and moving on down. Codon to anticodon brings in the amino acid. It's going to be peptide bonded to the next amino acid beside it. Then once it is, you can release that tRNA out and shift everything down and do it all over again, over and over and over again. So it's pretty cool. Again, the video is more elegant than this, but it is what's going on. In bacteria, they don't have a nucleus. So they have a severe advantage to our cells in a way. And it's why one of the reasons they can divide so quickly is they don't have to move their mRNA out of a nucleus. Their mRNA Doesn't need, it's already there where it needs to be. So they can actually start, and you'll see up there in that top picture. Look here, we have this DNA getting unwound. We're making our mRNA, the yellow guy, and we can bind our ribosome onto it before it's even done getting made. So before we've even finished making that mRNA, we can bind our ribosome on it at the same time and just start cranking down. And then just keep adding on ribosomes and adding on ribosomes and adding on ribosomes to keep making more of that protein. So this is called co-transcriptional translation. I always have to think through those words, but yeah, they can do that. We can't, because we have to get everything out of the nucleus. I think that's really cool stuff. Okay, so the COVID vaccine contains, viral mRNA, and I think I told you guys a little bit about this. But anyways, viral mRNA that we're going to be injecting into your arm, your muscle. And when your cells have that in there, it's just automatically going to start in with that whole process, the translation process, and start making COVID spike protein. And then your body's going to react to it. And it's kind of why you get all the weird like rash and everything that we saw and people who were reacting to it, that it will go away, because it's kind of attacking the surface of your own cells in a way. But once you get your antibody against that COVID protein, it's going to stop it from being able to infect cells. And if it can't stop it completely, which we know it can't, right? We know we can still get COVID, but it can make the ability of COVID to infect and to multiply and everything like that severely decreased. And we do know that because we can see the effect that it's had on society. If you guys think that COVID vaccines are a waste of time, I dare you to go back in time and see what it was like in the beginning, because people like me were dying. It was serious. It was serious business. So yeah, it works. It's just not 100%. It's not like, measles vaccine, right? So think about that. Well, probably why some of people got into medicine to begin with, but yeah, anyway, so when I talk about like where I went to school and everything like that and who I studied with and whatever, this is what I was talking about when I said that the guy that I studied immunology with developed this and has a Nobel Prize for it. Like this is the guy that I studied immunology with. So it's that stuff, which I'm, of course, proud of that. But somebody asked me the other day, was he a nice guy? And I was like, he was the guy. Like, I don't know. He was a guy. I mean, I knew him okay, but he was just there. I don't know, like every other professor, right? So anyways, so we can't do that co-transcriptional translation. We have to get our transcript, our mRNA transcript messenger out so we can do that. The AUG codes will be coding for different methionines, like the F-methionine is 4-male methionine. It has to do with the group that's added onto it and whatever. So I don't know. Also, bacteria can code for, they'll have that one promoter site, and you know how it will like be one mRNA for that one gene for us. For bacteria, they can promoter site and have several genes in a row. And they'll all be for different proteins, and then they can make a protein based on each of those from that one transcript, which is pretty cool. Which unfortunately is what we're going to talk about here in a little bit. All right. So for us, for the eukaryotes, we have something called introns and exons. Huge problem in genetic engineering, which we'll talk about in the next chapter. But introns are sequences that don't code for anything that we have to cut out and then join together the exons, which are the coding regions. So when we make our messenger RNA from our gene exactly, there's just junk in there that doesn't mean anything that we have to cut out. So imagine you're going in to make a copy of your, Smithsonian instruction on that gene. And it's just talking in the middle about how, and when I was little and blah, blah, kind of like stories that I tell or that Dr. Shearer will tell, okay? And we have to cut that out and make it about the information we're interested in. So the interesting part is the exons. The introns are the part where you're cutting out via a system called a spliceosome. It literally just pinches them together and cuts them off, like in the picture. That's really all you need to know about that. Next, we have operons. This is where the problems. So operons, these are exactly what I was talking about when bacteria can make one long mRNA that has several proteins in it. These are operons. We do not have these. These are only bacteria and archaea. Only bacteria and archaea. Okay. They can be inducible or they can be repressible. I want you guys to always remember what those words mean. They don't mean something different in this context. Because I feel like where people get lost is they don't know what inducible means or repressible means in this context. No, it means the exact same thing as it did before. It means the same. So when you're inducing labor, what are you doing? Causing labor to happen? Yes. If you were to repress labor, you would be trying to keep it from happening. It's not the term they use, but yeah, it would be the same concept. Yeah. Anyway, so that's the same terms. So inducible operants can be induced. They have genes that help, proteins that help metabolize nutrients. You're not going to see these nutrients every day. You don't need to have the genes on all the time. You only need them on when you see those nutrients. So it can be induced. The one that we learned, the first one about, I hate that, I forget that it has that in the order. But it's the lac operon or the lactose operon. The organism doesn't usually see lactose. So it's usually off. How is it off? What does that mean? Well, at the operator site, which is right near the promoter, at the operator site, we have a protein sitting there. Well, the repressor is repressor for both of them. They both have repressors, okay? To be clear, they both have repressors. But in the inducible, the repressor is already sitting there. And the polymerase, RNA polymerase, can't bind to the promoter and do its work because of it. It's blocking it, right? So when we see lactose, and we need those genes, lactose comes and binds to that repressor that's sitting there blocking, and causes it to change shape as proteins are wont to do when they bind with things, right? All those hydrogen bonds and all those charges and hydrophilic and everything like that, it's changing its shape by binding with lactose. Then it comes away, and now RNA polymerase can do its work. And when we run out of lactose, if we're not getting it in our diet anymore, eventually it will, I don't want to say degrade, but it won't no longer be bound to the repressor, and it'll go back to sitting there again. So this picture, somewhere, here it is. Okay. All right, so here's the idea. Don't worry about the regulator. This is where your RNA polymerase wants to make its mRNA, but it's not going to be able to work if we have this crap in the way. This is your repressor protein. It's going to sit there and block it. It's called locked. And we aren't going to make any of the proteins downstream. However, lactose comes in, changes its shape, it comes off, and now RNA polymerase can work and start making the proteins to help digest lactose. Lactose is transported and digested. Nice. Eventually, the lactose that started the whole unbinding will run out, and then we will sit back down and lock it back up again because we don't need it anymore. So it's inducible here. It was off. It had to be off in order to be induced. Why do we induce labor? Because the person is not going through labor and it's time, right? So this is not making our genes. It's not making our proteins from our genes. So now we're going to induce it because we need them. All right. This is the repressible operon. So basically it's always on. It's the opposite way, right? So we started off with the other one being off, and then turning it on when we need it. It's inducible. This is repressible. You can repress it because it's on. Okay? So it's able to be repressed. So it's on mode. It'll only get turned off when the nutrient isn't required anymore. Let me show you an explanation of it. So these are genes to help make arginine. Arginine is an amino acid. So if you're building a lot of proteins in the cell, generally you're going to be making arginine. This is the default, right? If the cell is using up that arginine like it's supposed to be, so we're making arginine because we're assuming that you need it. If it's using it up like it should, there's no problem. However, if the cell stops needing it, it's just not using it anymore. It's like a supply and demand type of a thing, where if we have a lot of that building up because the cell isn't using it anymore, now it is available to bind with the repressor. It's a repressor also, different kind, but it's a repressor. So now we have excess arginine. It'll bind with the repressor, and now that will bind onto the operator and block it. And that's how we turn that gene off. So it works kind of in the opposite. They're like opposites from one another, right? Inducible, we're going to be off, and you can turn it on, and then eventually it'll turn off again just naturally. This works the same way, but from on to off, and then it'll eventually turn on naturally when we need more. Okay. All right, now we have DNA recombination events. This is a recombinant. If I were talking about a recombinant, is any organism that contains and expresses genes that originated in another organism. Whether that is bacteria that are expressing insulin, or the corn that you're eating having genes in it from bacteria, or there's a lot of stuff. But that is all a recombinant. This could be naturally made or it could be scientifically made. Anyways, plasmids are one way that we can move from and create recombinants that are from species to species, creating a copy of our plasmid and then shooting it over to the next cell. And the way that it does that is through horizontal gene transfer. When we talk about vertical versus horizontal in genetics, we're talking about like your mother and father. If they pass something on to you, that's vertical transmission. If you have syphilis and you have a baby and you pass that syphilis onto your baby during birth, that's vertical transmission as well. Horizontal transmission is whenever you're getting something from another organism that isn't your parent, basically. So these guys are transmitting DNA between one another, that they weren't their progenitor cells. Okay, so they can do this usually using plasmids. Again, these are small circular bits of DNA that are not necessary, still considered part of the genome once they get in there. They're not necessary, but they do usually carry useful traits, things like antibiotic resistance. These are the three means by which you can achieve horizontal gene transfer. Conjugation, using the sex pilus, which is that little tunnel that we can make between the bacteria. Transformation, where like if bacterial cells near you die, you can take up their fragments of DNA. And then transduction is via the phage. So this is kind of what we're talking about with the lysogenic conversion. So you can achieve this by getting a phage from somebody. All right, cool. We did it, we talked about it. So there's a picture of transformation, just so you know. This bacteria died. That bacteria picked up its genes and put it into its genome. Cool, right? We're going to learn about that in the lab. So mutations are any change to nucleotide sequences in the genome. This is number one, the driving force of evolution. You typically have to have genetic mutations in order for it to be carried on. Otherwise, if just learning how to do a new thing, that doesn't get carried on to the next organism. So this can be done naturally through mutations that are just normal. You're making errors in the mutations, or it can be done by inductive reasons. Induced. So spontaneous is the one where it's going to happen randomly, just from DNA errors. Usually it's going to be DNA repair errors or DNA replication errors that are just getting missed and they'll get introduced, and they may or may not be helpful. Usually not, right? But if you're a bacteria, there's a video that I have somewhere. I think it's on, like, if you guys go to like the lectures, like the actual lecture part of it, it's mostly for the hybrid people. I don't know if you guys have ever gone through the modules, but if you click on like, this is the content for the course or whatever, it has a mini version of what I talk about in here. And there's some videos in there. I don't know why this version doesn't let me put videos in. I think it's because of just the rights that we have to use to sign into the computer or something like that. I don't know what it is, or if it's McGraw-Hill's thing. I don't know what it is. But anyways, this video is really cool, and I would encourage you guys to go see it. It basically is going to involve the concept of mutation happening spontaneously. So taking normal E. coli, some of us are working with E. coli in the lab, by the way. So taking that, putting it on a big table of agar, and over a week, observing. Now we'll put it at the end. There's no antibiotic, and then it'll be 1 time what we usually use to treat. 10 times what we usually use to treat, 100 times, and we went up to 10,000 times in the middle. And they're just normal. You're not encountering any other bacteria at all. They're just living there. And eventually, you get one mutation among those billions that are growing on there. That's all it takes, right? One. And now suddenly it's overgrowing into the area. And it does. That's exactly what happens. That's how easy it is to have antibiotic resistance. It's really important to remember that. Going into medicine and telling people to take their antibiotics the way they are supposed to. But anyways, induced mutations are whenever we have mutations that are coming from mutagens. A mutagen is anything that causes any genetic variation. It could be UV light. Okay, so that would cause potentially, if you guys don't know, we'll talk, I think, a little bit later about how UV light works to damage your DNA, but it literally damages your actual DNA code. So I'm just letting you know. Not that I'm going to put on sunscreen every day, but you know. All right, different kinds of mutations: missense, nonsense, silent, and back. These are the 4 of what we're interested in for single changes. If we are going from, I don't know, let's say it said, I know this isn't literal, okay, this is an example. If we went from UUA to UUG, and this was phenylalanine, and now this is leucine, I don't know. That is a missense mutation. Okay. Nonsense mutation is whenever you had UUA go into stop codon. It doesn't code for an amino acid at all, but it stops you. So if you had that, imagine if you had that mutation happen like on your second codon, you wouldn't get really any appreciable amount of protein. It could be a serious issue. All right, silent mutations. If you went from UA to, let's say, UC, and this was phenylalanine, and that was phenylalanine, they're different. It counts as a mutation, but it's silent because we didn't change the amino acid, right? Because we had some overlap. And then we have the back mutation, which is like if you had it change to leucine, let's say the UUG, and then later generations had it change back to the UUA, and they're no longer expressing that genetic deformity or whatever it was that happened from it. Yeah. Okay. It can be pretty severe, the effects of even just one change. So this is talking about just some examples of missense, a mutation. Anyways, I'm going to go over it. I kind of already gave examples. So frame shift mutations are worse by far, because what you have with a frame shift mutation, it's like if you were reading a book, let's say you had Harry Potter, right? And somewhere in there, there's a random word, and we're just going to delete 1 or 2 letters. And we have to shift every letter after that in the entire book up to that spot, but keep the same length of words. It would be ridiculous, right? Especially if you opened it way later on, you would not have any idea what was going on. It changes everything. It could lead to early stops. It can lead to a different, complete other protein being made, and it can be very serious for every gene potentially after that. So yeah, so that's the frame shift mutation. Bad. It's just some more examples of frame shift mutations. All right, then we have the Ames test. I think it's the last. Yeah, it is. The Ames test is a test to look at carcinogenic potential of certain things. So what they usually do is they have salmonella, and they turn this salmonella gene, manipulated its genes so it cannot make histidine. It has to be provided the histidine in order to live. And if they provide it the histidine, then it's fine. And if they don't, it shouldn't grow. If they expose it to a mutagen or carcinogen and it grows without them giving histidine, that means it got a mutation again. So now it can grow. So that's what the Ames test does. It looks for mutations in bacteria in certain situations where they couldn't do a thing and now they can. It has to be because of mutations. It doesn't happen spontaneously. Like, it doesn't come from nothing. It has to be a mutation of some kind. But yeah, that's it. So that's it for this chapter. We won't go through the questions again because this is a big chapter and I wanted to be sure we got enough of it in. But I'm hoping chapter 8 is usually not too bad. I'll say that if you are having problems understanding some of the stuff I'm talking about in chapter 6, try to at least look over it or look over some other resources that you have, because chapter 8 is all from this. Like, it's all going to be based on this. So I would try to understand it if you can. Okay, how? That's the power button. How is that not available? Okay, whatever.